Hello, and welcome to the best part of our microbial genetics lecture series in its protein synthesis, otherwise known as translation. We get to learn a foreign language today. You get to learn how to decode something. Okay, this is decoding the genetic code, the literal base pairs that we get in our DNA will make RNA. That RNA, as we saw, pairs to a tRNA. Okay, that tRNA carries one of these amino acids. Note how they are conserved, how UC and any other fourth nucleotide makes serine. Okay, same with proline. Okay, anything paired with CC read by a ribosome is going to give you proline. What do I mean by read by a ribosome? Well, it means it's a codon, or group of 3 amino acids. This codon must be in frame with the ribosome, right? If it's okay if we have UCU and we make a mistake, oops, I put UCC, that mistake isn't so costly and we're going to get serine, okay? Note we can get serine another way with AG and a couple of misplaced third point codon, okay? That is interesting because it shows that there is some variability allowed in the genetic code itself. And it's because we have all of these options, this whole table full of options, but only a certain amount of amino acids that can be made instead of all the combinations. So they pair up eventually at some point. Okay, and note here we have AUG, which means it codes for methionine. When you have methionine at the beginning of a template, that's actually going to start the whole thing off. It's a start codon, is what we call it. Okay. The ends falling off is called degenerate. That you can lose the third letter of the codon and it still be the same amino acid is degenerate. And just to clarify, on the far left is the first letter, C, right? And that's what's going to start that whole row. Everything is going to start with C in that row. In this row with my finger, everything starts with A because it's on the far left over there. And in this row right here with my finger, it's going to start with G because the G is all the way on the far end. Okay. U up top left is our second letter. So all the way down that column in the middle is going to be U. Second column all the way down is C. Third column all the way down is A. And fourth column all the way down is G. Okay. Then we go back to rows again. Everything on the third letter is going to be U on this row individually. Row C, row A, row G, all the way down. Okay. So that's how you'll read it. You will get a template strand and ATG, and you need to make that make sense in an RNA strand and then go from there. Okay, and remember, seeing these guys over here, if you have UGA and UAG, those are stop codons. Okay, what does stop codon mean? It means stop, don't proceed. The RNA polymerase will literally fall off. Okay, and if we have a stop mutation in the middle of our sequence, it'll stop right there and fall off, and you won't have access to that protein, at least made by that gene. Okay, pretty important. We'll get into that in mutations in a minute. When we do transcription and translation in prokaryotes, though, remember, as we talked about, it happens right away. As soon as this amino acid chain comes off, a ribosome hooks up as soon as it can and makes protein. Okay, and in eukaryotes this can't happen. It needs to come out of the nucleus first. We talked about transfer RNAs in the first couple of lectures, but we have 3 different models that you will see, and if your codon is GCC, your amino acid code will give you, will pair up right here. Okay, and it'll give you an amino acid that will be put on your peptide bond sequence. Okay, they transfer the amino acid to the growing peptide chain, as we talked about with the ribosome now for a few times. We need to make sure that we get it. Initiation is going to start it. Starts in the middle, then another one comes in and adds to the chain. Okay, another one comes in and adds to the chain, and when a release factor known as the stop codon gets there, the whole thing will fall off, and it won't be bringing another amino acid to add on, and so it tells it to go by. Okay, and again, if that's a mutation, that could be potentially deadly, and that the person is never even born. Keep talking about peptide bonds. Why do we call it peptide bonds? Well, because it's a bond between a carbon and a nitrogen, but specifically between this carboxyl group and this nitrogen with the hydrogen on it. Okay, and so this will join together enzymatically in the ribosome. Okay, brought together by a tRNA and the growing strand. Right, and then we have prokaryotic and eukaryotic protein synthesis. We've seen this picture now again. In prokaryotes, the process of transcription and translation occur simultaneously, and the nucleus separates these processes as needed. Okay, so let's test ourselves. Now that we know how to transcribe, we know what a complementary DNA sequence is, and we know how to translate, we can put it all together. All right, so let's get into it. So when we pair the complementary strand with the template strand, we just want to pair up the thymines and adenines and the cytosine and guanines. And we're going to pair those up according to Chargaff's rules, A's and T's, G's and C's. And it's just that simple. So our A becomes a T, our T becomes an A, our G becomes a C. The G's again becomes a C, 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 G, T, 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 A, C, G, etc. That is the complementary strand. It's pretty straightforward. Remember, in RNA, we switch our thymines for uracil. Okay, remember to switch those mentally. And we are going to also complement our template strand as we make it. Remember that RNA polymerase just makes a complementary strand essentially out of RNA on the template strand. Okay, so you'll see the trick as I point out after we finish the sequence, but let's start with ATG. Okay, ATG makes our mRNA sequence not thymine but uracil. Then thymine pairs with adenine, and guanine still pairs with cytosine. Okay, so then we keep going. CCG pairs with GGC, and then instead of TTT for adenine, 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 we're going to go uracil, uracil, and uracil. All right. Okay, and then another T. It's going to pair with an adenine, and then the C and G pair together. And you can keep that going and see how that works, and keep it going all the way throughout, and make that amino acid sequence before I get to the next page with the answers. Okay, and now we are going to send this out of our nucleus, and a ribosome will come and attach, and we'll start translating into an amino acid sequence. And any question on the exam, you'll have this available to you. You won't have to memorize this. What you do have to know is how to use it. Okay, and so we're going to make codons. Remember, codons with 3 amino acid sequences, or 3 nucleotides that will correspond to 1 amino acid. All right, so let's get to it. We're going to start with the first letter right there, second letter up here, and then the third letter right here. So big row, column, little row. Okay, so UAC is our template now. We have to go to our mRNA sequence because this is what got sent out. UAC is tyrosine. So I'm going to write TYR, tyrosine. Okay. CCG got sent out. So C, and G is proline. Okay. Proline. Our UUU got sent out of the nucleus on the same strand now. This is all the same strand. The ribosome is just coming and reading them all, codon by codon. Okay, UUU is right here, phenylalanine, PHE, and you can kind of get the picture here because ACG is going to make ACG. It's going to make threonine. Okay. All right. So continue the pattern and pause it here if you need to, and go ahead and try to work those out on a piece of paper in your head, wherever you need to before proceeding. So here's the answers. As you can see, our complementary strand all the way to completion. Our mRNA sequence is just like our complementary strand, except with U's instead of T's. And then our amino acid sequence, use this chart to point everything out. Note this stop codon here. Okay. If you see that on the exam and you get one of these 3, you'll put a stop and you just choose that answer if it corresponds to one of those. Don't worry about what it implies. Just go ahead and write the answer as a stop. But we are about to get to what it implies in the next video. This implies a early mutation in its sequence. Okay, and if it's a stop sequence right there, the ribosome will fall off right there and will no longer produce what's on the rest of our protein. Okay, and so right here we have a problem called a mutation here. Okay, and it is most likely a point mutation and an error in the last nucleotide. Okay, so probably went from an A and it should be a C. Okay, or maybe even a U. So that is what's likely, and go ahead and watch the next video as we get into what mutations actually are.